Hey, so good evening, everyone. Thanks for being here tonight. I'm Juliana. I'm assistant professor and beef extension specialist with Oregon State University. It's a great pleasure to be organizing these webinars. This is the second edition of this webinar series. Our first one was last year, and we're following up this year with the second edition. Our main goal with this webinar series is to provide information, discuss about wildfires with focus on preparedness prior to fire season and how to care for livestock during and after wildfires or during smoke exposure. This webinar series are part of a larger project funded by USDA NIFA, which with focus on wildfire smoke exposure and how that impacts animal health and performance, with major focus on beef and dairy cattle. As I said, this is our second edition, and for tonight, webinar session, we have Dr. Amy Skibiel and Dr. Pedram Rezamand from the University of Idaho. They're going to be presenting a little bit on their work on the dairy side of the things, on what they are seeing with dairy cattle when they're exposed to smoke exposure from wildfires. So Amy and Pedram, thank you very much for joining us tonight. I think you guys have your slides up, so I'll let you guys take the screen. Thank you.
Go ahead, Amy, if you want to introduce yourself.
Hello, everyone. I'm Dr. Amy Skibiel, and I'm a professor at the University of Idaho. Largely, my area of expertise is in lactation biology and environmental physiology, with interest in how animals respond to changing environmental conditions.
Hi, everyone. I'm Pedram Rezamand. I'm a professor of dairy nutrition with the University of Idaho Animal Science Department. My area of expertise is dairy nutrition with the focus on animal health and nutrient metabolism interaction. So for tonight's session, we're going to go over some of our research findings. Before doing so, I'm going to go over some of the background information and what's available in the literature right now. And then Dr. Skibiel will lead the discussion with our updated results. Over the past few years, we have gained a lot of new knowledge in terms of dairy and impacts of wildfire on dairy. So if we can move to the first slide. As you can see, we have a serious concern with regards to wildfire. Over the past several decades, they have become more frequent and more severe. The acres burned has gone up, and the areas that these affect have been increasing consistently. There are at least 7-8 large fires in California past few years. The average over the past 10 years has gone up, as you can see on the map on the right panel. Major areas of impact include the western states, and that's of concern simply because this is where we have a lot of dairy production. Basically, with the global climate change, this is going to increase the number of wildfire, and these happen because we probably have earlier spring snow melt and higher temperature, and the severity is also going to change because of extended wildfire season and the abundance of fire fuels. The length of the season also is going to change. With increasing fires, we have a greater chance of adverse health impacts from poor air quality, both on the human side as well as the livestock. As you can see, there's a specific geography relative to these wildfire trends. On the right panel, we can see that on the east side of the country, we have two lines here. The orange line shows 80s and 90s up to 2000, and the blue line shows 2001 through 2017. The wildfire trends are relatively slow. If you look at the area that is burned in terms of acres per year, they're pretty small. But if you look at on the left panel, the west side of the country, we can see that these are significantly different than the east side. Earlier in 80s and 90s, we have had up to about perhaps 0.6, 0.7 million acres burned per year. But last 2 decades, from 2001 onward, these have increased significantly, as you can see on the left side, the blue line, approaching 1.6, 1.7 million acres per year burned because of wildfire. So this is of a major concern for the western states. Why is it important for the western states? Because the majority of the industry happens to be on the western side of the country. USDA industry, if you look at it by region, we can see that we can see that on the left panel, total number of dairy cows by region, and western side is a massive. And if you look at the number of cows on the left panel, we can see that it's approached about 3.5 million dairy cows, milking cows on the western states, producing almost 50% of the milk production across the country comes from the area that we just talked about in terms of wildfire sensitivity. So beyond the dairy cows, we also have to take into consideration the number of calves and the system that we have here for raising those calves. About 1.7 million heifers are being born each year that are raised and maintained in production system in western states. And these calves that are born in these states are more susceptible to pollutions from wildfire, especially because many of these calves are housed in open houses and open barns and outdoor hutches with no particular ventilation system or protection. So they're exposed to whatever conditions that are outside, including wildfire smokes. And this makes the issue much worse, not only for milking animals, but also for calves. Moving on, we're going to talk a minute about particulate matter. The structures that are burned, land that are burned, produce a lot of toxics, and wildfire smokes include many of these air pollutants that are of concern with regards to public health, such as ozone, carbon monoxide, nitrogen dioxide, polycyclic aromatic hydrocarbons, and volatile organic compounds, and of course the particulate matters. There's a couple of different types of them. PM 10, those are the particles that have a diameter between 2.5 and 10 micron, and these are considered coarse particles. They're formed largely through mechanical processes and uncontrolled burning, demolition, residential burning, wildfire, construction, things of that nature. And then we have the fine particles, PM 2.5. These are particles that are smaller than 2.5 micron in diameter. They're formed largely through combustion and atmospheric reactions of gaseous pollutants. Of course, there is one smaller than that, ultrafine particles, but we're going to focus on PM 2.5. They're inhaled into the lungs, and they can deposit in lower airways and alveoli, and they have impacts on pulmonary system, local and systemic impact. So EPA has established a national standard for exposure. Current standard for outdoor PM 2.5 exposure over a 24-hour average is 35 microgram per cubic meter. So that's basically not appropriate for unhealthy or sensitive groups. Moderate is considered somewhere between 12 and 35 microgram per cubic meter. So what are the effects of wildfire particulate matter in humans? We can look at this set of effects in three major categories: pulmonary effects, cardiovascular effects, and mortality. There are numerous evidence that point out the detrimental impact of wildfire particulate matter on pulmonary system. Basically, diseases like pneumonia, bronchitis, asthma, and COPD have shown to increase in response to increasing PM2.5, especially on the smoky days. And of course, there are some evidence that support similar observation with cardiovascular health. Of course, not all of them point toward the same direction, but there are some evidence that show association between cardiovascular hospitalization, cardiac arrest, congestive heart failure, chest pain, and things of that nature related to particulate matter. We have to also note that certain populations are more vulnerable, including young, elderly people, and those with pre-existing conditions. So these effects seem largely attributable to two major issues here. One is inflammatory responses, and two is the oxidative stress. And this oxidative stress could be seen both locally and systemically. So we've had some studies in literature published in the past decade or two. Bring some example here just to familiarize the audience with the existing literature out there. For example, captive bottlenose dolphins in San Diego Bay were exposed to wildfire smoke in 2003 and 2007 when blood samples were assessed in terms of hematology. Few interesting findings were observed. Dolphins had higher carbon dioxide in blood during wildfire, and a month later, they also had a change in blood cell percentages, specifically increased eosinophils and neutrophils, part of the innate immunity during the wildfire as well as a month after the fire. And basically, these will tell us that wildfires have an effect on dolphin immune and lung function. Similar observations have been seen in other species, including horses with airway inflammation, and orangutans where energy metabolism as well as behavior were changed. In terms of energy metabolism, this is a very interesting observation. They moved into a negative balance, most likely because there was some type of shift in fuel and repartitioning of the energy towards a heightened immune response during this area. Now, I'm going to take a minute and talk very briefly and generally about bovine pulmonary system and why this is important. Cattle are specifically susceptible to respiratory diseases because of their unique anatomical and physiological properties of their pulmonary system. This could basically make them more sensitive to any type of air pollutants. They have fewer pulmonary capillaries per alveolar. They have lower lung compliance and greater resistance. They got longer trachea and bronchi. They have lower amount of lysozymes. That's part of their immune system. And they also have lower alveolar macrophages, which is also part of their innate immunity. Therefore, we strongly believe that cattle are particularly more vulnerable to inhalation of particulate matters, and sensitive to those health issues. So what are the effects of particulate matter in cattle? Exposure of cows to PM10 was associated with higher risk of mortality in short and long term in Belgium. This was not, of course, a wildfire, just ambient pollutant. Air pollution that related to ozone increased the risk of calf mortality in another study. Again, not related to wildfire, but just ozone. And particulate matter from dust on farm also was associated with elevated neutrophil counts in beef and dairy calves that were exposed, and these were higher in bronchioles of lungs and animals with pneumonia. So we have also had some preliminary study across several years and various farms in the Pacific Northwest, and Dr. Skibiel will go over those results. As I mentioned earlier, certain populations are more vulnerable, especially when you look at the young mammals, humans, or other species. We can see that young children are more vulnerable group because they have immature organ system and their immune system is not fully functional or competent, I should say. Same could go with elderly and those with pre-existing conditions. There are critical windows of developmental sensitivity to these environmental challenges that may have long-term impact on performance and health. For example, in mammals, the pre-weaning period, specifically in dairy calf, is one of those periods. Calves are born with an immune system that is naive and very much depend on passive transfer through colostrum feeding. So if there's any challenges in those periods, they will have a very different response, and they're more vulnerable to those environmental challenges. Average PM concentration in one study that was reported relative to calves with pneumonia was much less than what we see in wildfire smoke. This is of importance because when they're exposed to wildfire smoke, the PM concentration could reach a much higher degree, and that could pose a lot more dangerous situation. The effects are not limited to postnatal, but we also have evidence that would support the impacts in utero. So there's a greater risk in terms of human babies for preterm birth when they're exposed. There's a lower birth weight with newborns, and in animal model, evidence shows a greater inflammation, lower blood cortisol, and deficit in cognitive systems. So up to this point, there was no research in livestock, specifically in dairy and beef. And so Animal Science Department at the University of Idaho started work on dairy and in association with other universities that focused on other aspects. So the research projects that Dr. Skibiel is going to share with us include three large areas of coverage. First, lactation cows. Basically, our question is, does wildfire PM exposure changes metabolism, inflammatory responses, and production? The second project covered pre-weaned calves, and the main question we asked was, does the wildfire exposure affect behavior, metabolism, or health of these young calves? And then the third project, we just wrapping up now, is the impact on in utero. Are there any carryover effect? If the cow is exposed, is this going to have an effect on their baby once the baby is born? And with that, I'm going to ask Dr. Skibiel to please present the results of these projects. Dr. Skibiel?
Thank you, Dr. Rezamand, for setting up so nicely the rest of the presentation. So as Dr. Rezamand mentioned, these are the 3 research projects that we've been focusing on over the past few years, and I'm going to share some of the results from those projects. So we'll start with the first one, which is on lactating cows. And we did find in that study that milk yield was reduced when cows were exposed to elevated particulates from wildfire smoke. So this panel here just shows the decreasing milk yield as PM 2.5 on the x-axis is increasing. In this particular summer, we had concentrations of PM 2.5 reaching about 300 micrograms per cubic meter. And if you recall back to earlier in the presentation, when Dr. Rezamand mentioned the national standard for human outdoor exposure over 24-hour period, he had mentioned that number was 35 micrograms per cubic meter. So you can see that during a wildfire smoke event, those particulates are pretty high. And in fact, they can get even higher than that, up to about 500 or even 600. The lag day 0 means that this data was taken on the first day that the animals were exposed to that wildfire smoke. But we also looked to see if there were effects continuing onto subsequent days after exposure. And so this lag day 7 just shows 7 days later after the first day of exposure, is there still an effect? And you can see that, in fact, yes, there is. So there's still decreased milk yield 7 days after that first day of exposure. This last panel all the way on the right shows the change in milk yield associated with every 100 micrograms per cubic meter increase in PM 2.5 across all of the lag days. So you'll notice that on lag day 0, there's a decrease of about 1.2 kilograms per cow per day of milk. So that's roughly about 2.5 pounds of milk per cow per day. Going up to lag day of 7, we see decreases of about 1.5 kilograms per day, so roughly around 3 pounds per cow per day. Again, this is for every 100 microgram per cubic meter increase in PM 2.5. So when we have these really high PM days due to wildfires, like upwards of 300, then all of these numbers would need to be multiplied by 3. We also saw slight decreases in milk protein concentration with PM exposure as well. In cows, we also looked at effects on metabolism, knowing that temperature and humidity also has huge effects on cattle production and metabolism. We also included THI, which is this index of temperature and humidity, in our statistical models so that we could see if PM or THI or a combination of both were having an effect on the variables of interest. So on the previous slide, we saw that PM 2.5 alone could influence milk production. Here, we found that PM and THI together, when they're both increased at the same time, there are multiple effects on metabolism, including a decrease in blood urea nitrogen, suggesting lower protein catabolism. We saw increases in non-esterified fatty acids, suggestive of increased lipid mobilization, and we see decreases in body weight. We also analyzed hematology so we could look at the different immune cell populations in the blood, as well as red blood cell counts and hemoglobin. And we saw decreases in red blood cell count, in hemoglobin, but increases in eosinophil count and basophil count, which are 2 different types of white blood cells, and a decrease in neutrophil counts, again, with an interaction between PM and THI. So these effects are seen when both PM and THI together are elevated. So when we have a warm summer with wildfire smoke exposure. Interestingly, we don't see these effects until lag day 3. So the effects of PM exposure in THI on hematology are delayed. They don't occur for a few days after that first day of exposure. So just to kind of summarize the main results, obviously I'm not presenting everything we've found because we are limited on time, and I do want to have a chance to get to the other projects as well. But just to give an overview summary of what we found in terms of lactating cows, we did find that wildfire smoke impacts production, metabolism, as well as the immune system. So again, when PM 2.5 is elevated from wildfires, there's a decrease in milk production. It can be upwards of about 9 pounds per day per cow, depending on how severe the particulate matter concentration is in the atmosphere. And that's independent of THI. We found immediate effects of PM and THI together on metabolism. I didn't show the results for this, but we do see increased respiration rates when PM and THI are both elevated together, and delayed effects of PM and THI on blood hematology, including higher eosinophils, which could be a sign of an infection or an allergic type response, and a decrease in neutrophils. We might see this decrease in neutrophils if there's an immune suppression that's occurring due to wildfire smoke exposure, or perhaps the neutrophils are migrating to the lungs, which is the original initial site of those particulates entering into the body. So we've conducted some follow-up studies after that to try to get at what's happening there, and I'll explain some of those a little bit later on. So looking at some of the data that we have in calves, so these are calves that were exposed to naturally occurring wildfires in the postnatal period prior to weaning. So these are pre-weaned calves. Again, we do see that there's this interaction between PM and THI on metabolism, whereby when both PM and THI together are elevated, there's an increase in blood glucose and non-esterified fatty acids. And we see a decrease in the blood of beta hydroxybutyrate, one of the ketone bodies. Looking at hematology, we found some changes in hematology that were similar to the cows. For example, a decrease in neutrophils. We see a decrease in white blood cells in calves, and also a decrease in eosinophils and hemoglobin. I'm not going to get into what the acute phase response is here. What's important is that the acute phase response is part of inflammation, and there are a few different markers that you can look at in the blood to indicate when an inflammatory response is occurring. So serum amyloid A and haptoglobin are two proteins that are released into the bloodstream when there is inflammation occurring. So they're part of a pro-inflammatory response. And we do see changes in both of these markers in calves when they're exposed to increases in both PM and THI. We have used ultrasounds to be able to look at the lungs, and we can see where there are areas in the lungs where there's fluid where air should be. Consolidations could be an indication that the lungs have been damaged, or that there is an infection, or the calf is recovering from an infection. The problem with consolidation is where there is fluid, that limits gas exchange. We want to see nice aeration in the lungs, these nice pink lobes that you can see in this photo here, because that's where ample gas exchange is going to occur. In areas where there's consolidation and those air sacs are filled with fluid, there's limited gas exchange. And that is shown in this photo by these kind of darker brown areas. So that's indicative of lung scarring and damage. There are some studies showing that when calves have consolidation early in life, that can affect their milk production in their first lactation. I'm not going to go too much into the scoring system, but basically we use the ultrasound. We look at all the different lobes in the lung, and then we assign each of those lobes a score based on how severe or the consolidations are if there is a presence of consolidation. So a 0 means that there's really no consolidation. It looks like a normal lung, whereas a score of 3 to 5, for example, means that there's pretty severe consolidation occurring within that lobe. What we found is that, again, when PM and THI are both elevated together, we see an increase in these ultrasound scores, meaning that we are seeing more consolidation in the lungs. And these are occurring across lag days. So these are persistent effects that we're seeing across time after exposure. If we look at the proportion of calves that we did this ultrasound technique on across the study, you'll see the results here in this table. Before a smoke event, so these are baseline measures that we took prior to any smoke occurring. So in like the month or so leading up to a smoke exposure, we had no calves that had any indication of consolidations in the ultrasounds. During smoke exposure, there were close to 6% of the calves that were showing consolidation. But then in the week to 2 weeks following smoke exposure, about 12% of our calves had consolidation. We then looked at cell populations in the lungs. As I mentioned earlier, we noticed in cows that neutrophils were lower in the blood when they were exposed to smoke, which is not what we were expecting. We were expecting if the cows are mounting an immune response, that neutrophils would be increased in the blood. So we thought, well, maybe they're going to the lungs, the site of inhalation. Interestingly, though, we saw a decrease in neutrophil percent in calves that were exposed to wildfire smoke relative to baseline. So if you look in the figure on the slide, the neutrophils are shown here in pink, and you can see that for the baseline samples compared to the smoke samples on the right, there was a decrease in the proportion of cells that were neutrophils in the lungs, whereas there was an increase in macrophages in the lungs, as indicated by the purple bars. So during smoke exposure, there were more macrophages in the lungs than baseline prior to smoke exposure. And as Dr. Rezamand mentioned earlier on, those macrophages are really important immune cells in the lungs. They help to take up and break down particles and other foreign bodies and microbes that enter into the lungs. Some of these results we've seen kind of go hand in hand with the health scores that we observed. So we use this McGurk scoring system that is pretty standard in terms of doing an objective sort of calf health scoring metric, where you look at various physical attributes of the animal and assign a score to them. For example, the amount and the color of nasal discharge, the discharge that's coming from the eyes, fecal scores, so you can look at the consistency of the feces. And then cough scores, how much are those animals coughing, both with an induced cough and then spontaneously as well. And what we found is that, again, together, elevated PM and THI was associated with increased eye score, so there was more discharge coming from the eyes, and an increased cough score. Increases in these scores mean that that animal is showing signs of being less healthy overall. We've also conducted a behavioral study, which is in press right now, and where we just looked at the proportion of time that the calves spent either standing or lying. In this particular summer, we had 2 different wildfire smoke events. They were both about 3 to 4 days in duration and roughly the same magnitude. You'll notice that event 1 isn't shown in the green bars and event 2 in the blue bars. So just looking at the panel on the left, the time spent standing, during event 1, the calves were spending less time standing, more time lying, which you'll notice on the right figure. All right, during event 1. During event 2, we see a similar pattern of decreased proportion of time spent standing and an increased proportion of time spent lying. However, the magnitude of those changes was less during event 2 than it was during event 1. So perhaps the animals were becoming habituated or accustomed to that change in particulate matter, so that when event 2 came around, they didn't show quite the same magnitude of responses that they did during that first event. So just to kind of summarize, this is a picture snapshot of what we have found thus far is happening within the calf body when it is exposed to wildfire smoke during the pre-weaning period. I didn't mention these data, but we do see an increase in respiration rate and in heart rate and rectal temperature. And this is problematic because if the animals are breathing harder and faster, they're going to be taking more of those particulates into the lungs. We see alterations in immune cell populations, both in the blood and within the respiratory tract, which is indicating that there is inflammation around the whole body, but also locally within the lungs. We see an acute phase response, which is indicating that there is inflammation occurring. And we observe shifts in energy metabolism. So this could potentially have effects on animal growth, and we're looking into that now. We also found with our health scoring system that there were signs of eye and respiratory irritation because we see elevated eye scores and coughing scores. So again, overall, we see both systemic and local inflammation and overall effects on animal health and respiratory performance in terms of coughing and pulmonary consolidations. All of these things could potentially set that animal up for more infections, potentially from pathogenic bacteria, for example. This is a hypothesis that we intend to look into in the future. So just to touch on some of the work that we're doing currently, we are, as Dr. Rezamand mentioned, conducting a study to look at any potential effects on that postnatal calf from exposure during gestation. So in this case, we had a control group of cows that were not exposed to wildfire smoke during pregnancy, and we had a group of cows that were exposed to wildfire smoke during the same time during gestation. And we've found some interesting results thus far, but again, this is a work in progress. So we are still working on analyzing the data from this project. So one interesting finding was that body weight, which is the panel in the middle here, and I should point out the green bars are the control non-exposed group, and the orange bars are the smoke exposed group. So again, these data are showing the calves postnatally from exposure prenatally. So for body weight, we found that our wildfire exposed group were lighter at birth relative to our control group, but by day 14, we didn't see any differences in body weight between the two groups, suggesting some compensatory growth in our wildfire smoke group. This can be seen with the average daily gains as well. So we looked at average daily gain both pre-weaning, so from birth to 8 weeks, and then also in the post-weaning period from about 8 to 13 weeks. So in the pre-weaning period, our wildfire smoke calves had a greater average daily gain relative to our control group. But in the post-weaning period, we see the opposite effect occurring, where the control group actually had higher average daily gains relative to our control group, or sorry, relative to our wildfire group. And both of these groups were fed the exact same diet on the exact same feeding protocol. We also found that our in utero exposed group had higher health scores at 14 days of age. Again, higher health scores are sort of indicative of a slightly less healthy calf. We found increases in eye scores and navel scores, potentially, again, indicative of an inflammatory response, and an increase in fecal scores, all in our wildfire exposed group relative to the control calves. We kind of see similar effects of wildfire smoke exposure on respiration rates in our in utero exposed calves. So the wildfire exposed group had higher respiration rates at birth relative to our control group. None of the cows on the trial had any issues with calving. By 14 days, we still see differences between our treatment groups, but now our wildfire smoke group actually had a lower respiration rate relative to the control group. We also measured cortisol just as an indication of stress and also as a metabolic marker. And this was not a significant difference, but there was a tendency for our wildfire smoke exposed group to actually have lower cortisol concentration in the blood relative to our control group. So what does all of this mean? To kind of summarize our in utero observations, we find that health scores were different between our two groups at 14 days of age. We did not really see differences in health scores at birth, though. So it is possible that there's some environmental effect that was occurring after birth that's contributing to that disparity at 14 days of age. We can't entirely rule that out, although they were managed in the same way, and we tried to control any other environmental exposures as much as we possibly could. The lower cortisol at birth in our exposed group is kind of interesting. We're not entirely sure what this means in terms of an immune function or metabolism, but there is a study in monkeys that found a similar effect of infants that were exposed to wildfire smoke early in life. Regardless, there seems to be lingering effects of wildfire smoke exposure in utero on that calf. It's possible that particulates can cross the placenta, or they're contributing to oxidative stress in the cow, and that's impacting potentially fetal growth, for example. There might be changes in blood pressure or nutrient supply to that developing fetus as well. Not entirely sure what the physiological mechanisms are that are contributing to the postnatal impacts of prenatal exposure. Those are all great questions to address in the future. And certainly knowing that exposures to other adverse environmental conditions prenatally can have long-lasting impacts on the postnatal calf. It would be very interesting to follow these animals long-term to see if there are any implications on their survival or future performance later in life from this early life exposure to wildfire particulates. So just to kind of wrap up what we talked about in this webinar, wildfire particulate matter, either independently or along with elevated temperatures, has an impact on calf behavior. It changes the way that cows and calves partition and use energy. There's a decrease in milk yield. There seems to be ample evidence for inflammation, both locally within the lungs and systemically across the body. And there seems to be an effect on growth in utero, and that's indicated by the differences in birth weights that we see between our two treatment groups. So I'm not really going to go too much into detail on here on this slide. Because there's going to be a whole webinar dedicated to tips and tricks and guidelines for protecting livestock from these adverse effects from wildfire smoke. That will be on Wednesday. And I've also left here the website livestockwildfirehub.org, which collaborators at University of Idaho, including Dr. Rezamand and myself, and researchers at Oregon State University, including Dr. Ranches and Dr. Cruikshank, who are also on the webinar today, were involved in creating. And this website really has some great resources for producers and stakeholders to not only protect animals from wildfires when they occur, but how to set up a good plan and to have that plan in place to implement in case of an emergency. So I encourage you to check out that website. I've included the QR code here as well. And with that, I would just like to thank everyone that's been involved in these projects. And all of the collaborators across institutions, including Oregon State, University of Idaho, South Dakota State University, the commercial farms that were involved, from which we sampled animals, and all of the students that participated in sample collection. And with that, I believe we still have quite a bit of time to answer any questions.
Yeah, thank you, Dr. Skibiel and Dr. Rezamand, for the presentation. I'm really happy that you guys had some time to share what you're doing, especially when we think about how big is the dairy industry in the states that are most affected with a wildfire. So it's just amazing to see all this data. And like with your last slide with acknowledging everybody, it takes more than a village to get all this data put together. It's not an easy subject to be studying it. So I'm really happy that you guys are doing it. I feel like there's a lot of questions that we still have to answer. Same thing on the beef side. We have some data that we'll present Wednesday that similar to what you're seeing on the dairy side. So it's exciting to see that we're moving on together on this topic. So it's really nice. I do know that there's a few questions on the Q&A box. So if you want to go ahead and maybe read those for us and answer, and this will also give time to other folks to type those questions if they have.
I can't see the chat.
Oh, I can read it for you then.
Yeah, perfect.
So one of the questions was, how was the induced cough test performed? I think that was when we're talking about cough scores and health scores.
Yeah, so this is standard. It's part of that McGurk health scoring system that came out of the University of Wisconsin. And basically, you take your fingers and thumb and you flank the trachea, and you just rub up and down a couple of times, a little vigorously on the trachea. And that's what we call an induced cough. So if the calf coughs after that maneuver, we would consider that to be an induced cough. And we count the number of coughs that occur after we do that maneuver. And then we also listen for any just spontaneous coughs without us even touching the animal.
That's pretty good, thank you. And the other question was, what were the average ages of calves in event 1 compared to event 2?
That's a great question. I think I'd have to go back and look exactly, but I believe in event 1 and event 2 happened within 1 week of each other. So I want to say they were somewhere around 30 days of age or so, 30 to 40 days. And then it was about 47 to 50-ish for event 2. So
it's probably still within the same phase of development, right? Like not they were like pre-weaning, that was totally post-weaning. They were still
exactly.
bleeding.
Yeah, exactly.
I do have I think that's where the questions we had on Q&A, but so we learned a lot today, and this is just a little bit of information so far, but still it's a lot. So what can we do with all that information? How can producers use all that information? And how do you guys see this projects moving forward?
Yeah, Dr. Rezamand, I don't know if you want to take a stab at this one and then I can add anything else.
Sure. Basically, we need to learn some of the mechanistic relationship that exist here. We need to learn the physiology behind the responses that we see when animals are exposed and post-exposure. And based on those, I think we can set up some recommendation in terms of nutrition, in terms of environmental conditions such as housing and ventilation and things of that nature. These are obviously ultimate goals, and we have long ways to go. Right now, what we're learning last year or so and moving forward the next year or two are the basics and foundation for reasoning behind what we see. We know what will happen now to young animals or when they're pre-weaned or early post-weaning. We know what will happen with milking cows and in utero effect. What we're doing right now is to gain a better understanding of why these things happen and what are the mechanistic relationship here. And based on those, there's good chance that we could make some recommendation in terms of inflammatory responses, some recommendation in terms of antioxidants, some recommendation in terms of hydration, and of course, recommendations in terms of housing and ventilation.
I don't think I have anything to add to that. I think Dr. Rezamand covered that pretty sufficiently.
Yeah, I agree, and I agree with all the points. We still have a lot to learn, and I'm sure all the data we are getting will lead us to come up with suggestions for producers on how to better prepare for those situations or recover from those. Do you guys want to talk about future directions, maybe like where you're gonna go? I mean, kind of touch a little bit on that, but if you want to add more.
Yes.
Go
Go
ahead,
ahead.
Dr. Skibiel.
I was just going to say one of the trials that we're going to be starting pretty soon is looking more, as Dr. Rezamand mentioned, into the physiological mechanisms, particularly within the lungs, to see, you know, why does wildfire smoke have these effects on immune cell populations? What's going on with their functionality? Thus far, we do see that there are some, you know, immune system changes, but we're not entirely sure how that affects immune function. So trying to get a little bit more into the functional effects is what we have going on in the near future.
Oh, that's excellent. That's exciting. We're discovering and learning every day a little bit more. I think that's all we had on questions. So just want to thank you both very much for joining us and being part of this group. I really appreciate the collaboration and everything that we do together. Thank you very much. I think what you guys are doing, it's amazing. And I think that's what we had for tonight. So tomorrow we're going to have Dr. Katie Wolstein joining us. She'll be talking about fire preparedness, how to be ready for those situations that we don't want to be involved, but better be ready than sorry, I guess. So I hope to see you guys tomorrow night. And thank you all. Thanks, folks. Bye.
Thank you.